Let us discuss interpretation of simple organic molecules from NMR spectra. Interpreting NMR spectra of simple organic molecules involve analyzing chemical shifts, integration, and splitting patterns to deduce structural information, including the types and numbers of protons and carbons present, as well as their connectivity. NMR interpretation plays a pivotal role in molecular identifications. As interpreting NMR spectra, the structure of an unknown compound as well as the known structures can be assigned by several factors such as chemical shift, spin multiplicity, coupling constants, and integration. NMR spectroscopy is a crucial analytical tool for organic chemists. The research in the organic lab has been significantly improved with the aid of NMR. Not only it can provide information on the structure of the molecule, it can also determine the content and purity of the sample. NMR spectroscopy analyzes atomic nuclei in a magnetic field, providing insights into molecular structure, functional groups, and bonding by analyzing chemical shifts, splitting patterns, and integration. Important concepts in the interpretation of NMR spectra. Chemical shift. The position of a signal on the NMR spectrum, measured in parts per million, reflects the electronic environment of a nucleus. Protons or carbon atoms in different environments will resonate at different frequencies. Reference standard. Tetramethylsilane is used as a reference with its peak at 0 ppm. Integration. The area under each peak in the NMR spectrum is proportional to the number of protons that resonate at that frequency in that chemical environment. Interpretation. By comparing the areas of different peaks, one can determine the ratio of different types of protons in the molecule. Splitting patterns, that is multiplicity. The splitting of a signal into multiple peaks, it can be doublet, triplet, or quadruplet, and so on, is due to the interaction of a proton with its neighboring protons. N+1 rule. The number of peaks in the signal, that is multiplicity, is determined by the number of neighboring protons plus 1. Singlet, that means only one peak. No neighboring protons when n equals 0. Doublet, there exists two peaks, one neighboring proton, that is n equals 1. Triplet, we have three peaks, two neighboring protons, that is n equals 2. Quadruplet, there exists four peaks with three neighboring protons when n equals 3. Example, a proton with two neighboring protons will appear as a triplet. Coupling constants J. The distance between the peaks in a split signal, example, the difference between the peaks in a doublet, which can be used to determine the strength of the coupling between neighboring protons. Proton NMR focuses on the hydrogen nuclei, that is protons, in a molecule. Protons in the same chemical environment are considered equivalent and will give rise to the same signal in the proton NMR spectrum. Example, in ethanol, the three protons on the methyl group are equivalent and will appear as a single signal, with two protons on the methyl group are also equivalent and will appear as another single signal. Carbon NMR focuses on the carbon nuclei in a molecule. Methods of interpreting NMR spectra. 1. Count the number of signals. we should count the number of signals in the spectrum to determine the number of different types of protons in the molecule. The number of distinct signals in an NMR spectrum indicates the number of different types of protons or carbon atoms in 13C13 NMR in the molecule. Second one, analyzing chemical shifts. We should analyze the chemical shifts to identify the types of protons based on their environment. We should compare the chemical shift values of the signals to known values for different function groups and types of protons. Third one, determining splitting patterns. We should use the integration values to determine the ratio of different types of protons. We should identify the multiplicity of each signal, singlet, doublet, triplet, and so on, to determine the number of neighboring protons. Using integration values, we should use the splitting patterns to determine the number of neighboring protons. The integration values provide information about the relative number of protons or carbon atoms that resonate at each frequency. Combined information. We should combine the information from chemical shift, integration, and splitting patterns to deduce the structure of the molecule. By combining the chemical shift, splitting pattern, and integration data, we can deduce the structure of the molecule. Information obtained from proton NMR spectrum are 1. The number of signals indicate the number of different types of hydrogens. Second one, integration or peak area indicates how many hydrogen molecules are in each signal. It is given in ratio. Chemical shifts are given in delta values. The chemical shifts values indicate the electronic environment of the hydrogens, shielded or de-shielded. Splitting patterns indicate the number of neighboring hydrogen atoms. The magnitude of the coupling constants, J values, depend upon the spatial relationship of the two atoms. Examples, ethane. In proton NMR, ethane shows a single signal, it's a singlet, because all the hydrogen atoms are equivalent. Ethanol. In proton NMR, ethanol shows three signals, a triplet for the methyl group, a quadret for the methyl group, methylene group, and a singlet for the hydroxyl group. Propanol. In proton NMR, propanol shows two signals, a singlet for the two methyl groups and a singlet for the carbonyl group. The area under the peak of a proton signal is proportional to the number of equivalent hydrogen atoms, giving rise to the peak. The proton NMR spectrum of a compound with one methyl group, one methylene, and one methane will have three peaks with the peak ratios of 3:2:1. Thus, the proton NMR spectroscopy identifies different types of hydrogen atoms in organic molecules by analyzing their unique chemical shifts, integration, area under the peak, and splitting patterns, providing insights into structure and connectivity. Let us learn to understand the proton NMR spectrum and elucidate the structure of a compound, for example, methyl acetate, from proton NMR spectrum information. Generally, the information about the structure of molecule can be obtained from four aspects of a typical proton NMR spectrum: chemical equivalent and non-equivalent protons, that is total number of signals. The second one is chemical shift. Third one is integration, and the last one is signal splitting pattern. So here is the example of the proton NMR spectrum of methyl acetate. So we have three signals, and the first one which shows 0 ppm, which is nothing but your reference signal, which is nothing but TMS. And there are two signals exist, whereas in methyl acetate we have 3 oxygen, which is in C double O bond, and another 3 hydrogen atoms with C double bond with oxygen, and 3 hydrogen atoms with oxygen bond. These 3 are equivalent, and these 3 are equivalent. So this will produce one signal, and this will produce another signal. So that's why we have two signals. Actually speaking, we should have 6 signals. So this we have 3 hydrogen atoms here, which is bonded with CO here. Then another 3 hydrogen atoms, which is bonded with O. We should obtain 6 signals, but instead we have only 2 signals because these 3 hydrogen atoms are equivalent in CO double bond, and these 3 hydrogen atoms are equivalent with oxygen bond. So that's why we have 2 signals. In the above proton NMR spectrum of methyl acetate, in the above figure, we can see that there are 3 signals. The peak at the far right is for the standard reference compound tetramethylsilane. So the compound methyl acetate shows 2 signals in proton NMR spectrum, but totally it contains 6 hydrogen atoms. This is because of chemical equivalence. The total 6 hydrogens can be divided into 2 groups. The 3 HA protons in the methyl group that bonded with CO are all in the same chemical environment. Therefore, they are chemically equivalent. All chemically equivalent hydrogens have the same resonance frequency with applied external magnetic field. So it shows only one signal in proton spectrum. On the other hand, we have 3 Hb protons in the methyl group bonded with oxygen atom are chemically equivalent as well and show the other signals. That's why there are two signals for compound methyl acetate. The ability to recognize chemical equivalent and non-equivalent protons in the molecule is very important in understanding NMR spectrum. For the compound with structure given, we should be able to predict how many signals are there in proton spectrum. On the other side, if the proton spectrum is available for an unknown compound, counting the number of signals in the spectrum tells us the number of different sets of protons in the molecule. And that is the very important information to determine the structure of compound.